Good evening to everybody, and welcome to the fourth lecture of the 2025 edition of Horizons in Medicine and Biology. I am Lorenzo Della Casa, and this is my colleague Alessio Morosi, both second-year medical students here at Scuola Superiore Sant'Anna. Before we begin, here's a brief introduction for those unfamiliar with this institution. The Scuola Superiore Sant'Anna is a public university that admits students purely on merit through a highly competitive national selection process. Students at the school have the opportunity to tailor their university studies to their personal interests. They thrive in a dynamic, multidisciplinary environment where individuals from different fields come together to participate in shared courses and projects while living in the same community. This collaborative setting fosters the exchange of ideas and nurtures innovation.
Thanks to this event, Scuola Superiore Sant'Anna is thrilled to welcome distinguished experts in the biomedical field as one of us for a day. The host will join our community, sharing his expertise and journey with students and faculty alike. It is a unique opportunity to connect with such inspiring figures, underscore the school dedication to creating moments of meaningful interaction and inspiration. Students will gain invaluable insights not only into the latest advancements in medicine, but also in the professional path of leaders in the field. For one day, they become a part of us, of our academic family, leaving a lasting impact and sparking new ideas that will shape the future careers of many. This rich cultural environment led to the establishment of this lecture series. The seminar gave us the opportunity to host in our school many brilliant researchers devoted to medicine, who gave us little pieces of their great research experience. Before the start of this lecture, we would like to share with you a short summary of the past editions of Horizons in Medicine and Biology through this video.
Students like yourself are very impressive to me, and I can understand that it is a very strong selection.
La Scuola Superiore Sant'Anna è ovviamente accompagnata dalla fama dell'aver formato tante persone che ricoprono ruoli importanti nel paese.
That's something very special, it's something very exclusive and unique, and so immediately I say, I want to be there, I want to do this lecture, and I want to know the people there, how they work, because for me it was a surprise.
Per esserci stato in passato, ma anche per fama, perché la Scuola di Sant'Anna è certamente una delle istituzioni educative fra le migliori in Italia.
Orizzonti mi permette di conoscere e approfondire quelle che sono le ultimissime innovazioni nel campo della ricerca biomedica e di conoscere da vicino i protagonisti che fanno.
Orizzonti mi permette di esplorare temi che non sempre si affrontano durante i percorsi universitari e di capire come la ricerca e l'innovazione possono tradursi nella pratica, cosa che sarà fondamentale durante il mio percorso lavorativo.
Orizzonti significa per me futuro, ricerca e scoperta.
Internazionalità, cura e conoscenza.
Thanking Sarah Techman for being with us today, I give the floor to Professor Vincenzo Liunetti, the architect of this seminar, who will introduce our guest. Thanks.
So good evening and welcome to this new Lectio Magistralis Horizon in Medicine and Biology. It's a great pleasure to have here Sarah Amalia Teichmann, and actually she served as a senior research fellow at the Churchill College at Cambridge University, and she served as professor of stem cell medicine at the Cambridge Stem Cell Institute at the Department of Medicine of Cambridge. Professor Teichmann was educated at the European School in Karlsruhe in Germany, where she completed the European Baccalaureate in 1993. And then she went and studied the Natural Science Tripos at the Trinity College of Cambridge, and was awarded a first-class Bachelor of Arts degree. In 1999, she completed her PhD at the Laboratory of Molecular Biology on genome evolution using a bioinformatic approach. Following her PhD, Sarah Teichmann did postdoctoral research fellowship supervised by Janet Thornton at the University College of London, and she was a medical research council program leader later on, studying partnering in protein interaction and transcriptional regulatory networks. From 2005 to 2015, she served as a teaching fellow and director of studies at Trinity College of Cambridge. And in 2013, she was appointed the joint position as a senior group leader at the Wellcome Trust Sanger Institute and the research group leader at the European Bioinformatics Institute until 2016. She was also head of cellular genetics at the Wellcome Trust Sanger Institute until 2024. And from 2015 to April 2024, she was a director of research at the Cavendish Laboratory Physics Department, Cambridge University. But what she does? Sarah Teichmann investigates gene expression and the protein complex assembly using both wet laboratory and computational biology technique. She's a very interesting approach. And for the science that she produced, she won several awards. In 2010, she was awarded the Colworth Medal from the Biochemical Society, and then she was awarded the Francis Crick Medal and membership of the European Molecular Biology Organization, the Lister Prize from the Lister Institute of Preventive Medicine too. In 2015, she was awarded the Michael and Kate Barney Award for Young Investigators by the Biophysical Society and the EMBO Gold Medal. Professor Teichmann was elected a Fellow of the Academy of Medical Sciences in 2015, and the Helmholtz International Fellow Award 2 years later, and she received the Genetic Society Mary Lyons Award in 2018, and the Biochemical Society GlaxoSmithKline Award in 2020. While I was trying to summarize the so important CV of Professor Sarah Amalia Teichmann, I read that he has also been an activist for a woman career in science through enabling scientists in family to advance their careers while working part-time. She chaired the Sex in Science debate at the Wellcome Trust on balancing family life with working in research. And in 2016, she was elected an International Society for Computational Biology Fellow. In 2020, she was elected a Fellow of the Royal Society, and in 2023, she was a winner of the Federation of European Biochemical Society EMBO Women in Science Award. The last achievement is the Ramar Chair of the India Academy of Science recently in 2024. And she published over 394 publications, reaching an H index of 116 Scopus. And actually, she's member of the board of reviewing editors of Science since 2012. And she also has distinguished editorial activity in other important journals like Advanced Genetics and Developmental Cell. She is mother, 2 daughters, and it's really interesting to know that in 2022, Professor Teichmann collaborated in the production of an experimental film. The name is Constellations, which was produced by her sister, artist Esther Teichmann, and the visual artist Christopher Stewart as an exploration of the human cell atlas. But what is it, the human cell atlas? Please, Professor Teichmann, tell us about this. Thank you.
Thank you, Professor Leonetti, for that lovely introduction. Thank you for having me here and for hosting me. And I also want to say thank you to Lorenzo and Alessio for looking after me so well today. So indeed, what I'm going to be talking about today is an introduction to the Human Cell Atlas and to some of the applications for learning about development and disease. And yeah, okay, perfect, thank you. So the Human Cell Atlas has been buoyed by 2 major kind of categories of technologies. The first is the resolution revolution in genomics, which was single-cell genomics. And what that meant was that we were able to sequence the nucleic acid content of individual cells or individual nuclei. Whereas before, we needed hundreds or thousands of cells to have enough material to sequence DNA, RNA, et cetera, from a cell population. And so what the single cell approach allows us to do is to take a sample like a heart biopsy, sequence every cell and nuclei within the sample, and then analyze the data using a data-driven approach to classify the different cell states and the different cell populations. And so that allows us to then define cell states, cell types in an unbiased way and make new discoveries that are unexpected and surprising. And coupled with that resolution revolution, in terms of suspension cell analysis of cells and nuclei are the spatial technologies, spatial transcriptomics and spatial biology with highly multiplex molecular readouts that are either sequencing based or imaging based. And of course here what you can do is take a thin tissue section, a thin slice like a salami slice, and then map the sort of geography of the transcriptomic landscape of the individual cell, the tissue with the individual cells in a high resolution manner. And as you can imagine, basically taking the data from a matched tissue sample in terms of, for instance, having the suspension cell or nuclear data sets and having the tissue sections and integrating this with data science, with AI/ML techniques, gives us a detailed picture of a tissue section at unprecedented resolution so that we understand in detail, in deep, full molecular breadth, not only the individual cell types, but also where they sit next to each other in the tissue. And so, you know, buoyed along by the development of these technologies over the last 15 years or so, we got together, so Aviv Regev and I got together and decided to launch the Human Cell Atlas Consortium as an international initiative to create a comprehensive reference map of the types and properties of our cells, the fundamental units of life. And of course, this is a basic science project where we want to understand the nature of our cells and tissues at molecular resolution, but it's also a project that has medical implications for diagnosing, monitoring, and treating disease. And what I want to emphasize is that this is a global community. It's an international project that's a grassroots-led initiative driven by scientists for scientists, and it's open for everybody to join and become a member by simply signing up here at humancellatlas.org/join-HCA. So please feel free to visit the website. All you have to do to sign up is basically register and, you know, commit to the principles of data sharing, ethical principles, and so on. And what it then gives you is access to the newsletter, the meeting schedules, the communities, and so on, that how we organize ourselves. So this slide kind of shows the different institutes. These are the numbers of institutes, so 550 in Europe, 470 in Asia, and so on, that participate in the project. And now we are over 4,000 members. So this slide is slightly out of date. And the kind of short history of the project, as I said, is we were founded in 2016. We had a small meeting with about 100 members, and that sort of allowed us to enter phase 1 and start establishing the community and start generating data. We are now really firmly in phase 2, where we are integrating data for the different bionetworks of the human cell. So we organize ourselves into 18 biological networks, where each one represents one of the major organs or systems in our body. And the major activity that the community is working together on now is to produce draft data objects for suspension cell data sets. And the Lung Cell Atlas version 1.0 was published in Nature Medicine, and can be accessed at data.humancellatlas.org. Brain published in Science. Organoids has come out in Nature Genetics. The retina has also been published, and there are 14 more in progress. We just approved the kidney, the gut, so small and large intestine, development, and liver. And each of these kind of reference data objects, at least for suspension cell data, is posted with versioning at data.humancellatlas.org. And so you know, this will be an iterative process, just like the assembly of the Human Genome Project with the chromosomes, where the assembly was kind of in a sort of stepwise, you know, assembly of the individual contigs, the individual chunks, to make the so-called golden path of our chromosomes. We are now in the process of assembling the tissue and the cells in the tissues and organs in terms of their complete molecular fingerprint. And this, again, like the Human Genome Assembly, this will be an iterative project, and it will also go in parallel with expanding and completing the atlas. And I'll talk about that more, and will become more clear. So we are at over 100 million cells that have been mapped from 18 biological networks, and these images kind of represent the individual biological networks and the different organs. And I'm going to kind of stop there with the introduction and dive straight into work that we've been involved in our group over the years. And I'll start with really an organ that's at the beginning of the life of every single person, which is the placenta. So the placenta is, as I'm sure you know, embedded into the uterus, and provides basically the interface between the maternal blood flow and the embryonic or fetal blood flow, and delivering oxygen and nutrients basically across this boundary of two organs. So why were we interested in studying this organ? You know, that's transient, but it's absolutely essential for establishing successful pregnancy. And the reason was that I was interested in T cells specifically, and had worked on mouse immunity and mouse T cells, and started our first kind of single cell experiments on T cells, which are of course very easy to isolate from spleen. And the big mystery, basically, from a T cell point of view, is how does the maternal immune system avoid rejecting the placenta, which of course contains 50% paternal alleles. So roughly half the alleles in the trophoblast cells in the placenta are non-self, and basically the NK cells and the T cells should be recognizing those alleles as non-self in theory. And so the question that we were asking is, what are the cells and what are the tissue architectures that allow a peaceful coexistence of the maternal immune system and the trophoblast cells with the embryonic or fetal genome? How is that possible? And so the way we wanted to answer that question is mapping all the different tissues in the placenta, and you can kind of get an idea of the different regions. So you have trophoblasts, these purple are the trophoblast cells, the extravillous trophoblasts, which are budding off from the placenta and entering the uterus, the endometrium, which is called the decidua in the pregnant state. And then there are different trophoblast subtypes here, syncytial trophoblast, villus cytotrophoblast, and these are the fetal fibroblasts, fetal immune system, the Hofbauer cells, etc. So we set out to basically this was a really talented postdoc in the group, Rosa Ventotormo, who worked with another incredible postdoc, Mariana Efremova, to sample all the different tissues, disaggregate the cells, and then generate single-cell RNA sequencing data and analyze the data. And what this revealed was essentially the different subsets of trophoblast in unprecedented detail. We discovered new placental macrophages of maternal origin that are sitting around the edge of the placenta and patrolling the placenta. And this was the key thing here was that because we were sequencing the RNA in each single cell, we could tell whether it's of maternal or fetal origin. Whereas previously, without having the genetic resolution data, it was unclear when these cells mix, which cells are from one genome and which cells are from the other genome. And so what that told us was that these macrophages that are patrolling the edge of the placenta are actually maternal and not fetal, which was a surprise. We were able to define different NK cell substates. So you can see the three different green colors here reveal different activation states of specialized so-called decidual NK cells that are kind of in a tolerogenic state and not activated for killing and rejection. And the mechanisms for those tolerogenic cell states are shown here, where we are interpreting the transcriptomic data in terms of the cell surface receptors. So what are the proteins that are on the cell surface of these natural killer cells of the maternal immune system? And how are these cells kept in a peaceful state by interacting with the fetal trophoblast cells and also with the maternal stromal and macrophage cells, so represented schematically here. And the key, basically the clue, lies in the kind of intercellular conversation that's taking place between the cells, so the ligands and receptors at the interfaces at the cells and that are controlling the signaling and keeping this tissue microenvironment, these tissue niches, in a state where the uterus is remodeled for increasing the blood flow to the placenta, increasing the size of the arteries, but basically not killing. And you can see here on this NK cell state here, there's an enzyme that's metabolizing ATP and reducing inflammation, CD39. There's a very special fetal HLA here, which is HLA, as you know, is the molecule that presents antigens to the immune system and allows us to distinguish self from non-self. And this special HLA basically contributes to sort of hiding the paternal antigens, if you like. And then there are a lot of other receptor ligand interactions that are involved in maintaining that peaceful interface. And that really, advanced our understanding of how the remodeling of the uterus works in first trimester pregnancy. So these are first trimester placenta from social terminations, from the Human Developmental Bioresource that's in London and Newcastle, supported by Wellcome and the MRC over the past 20 years. A very precious tissue resource. So how did we infer those receptor-ligand interactions that I just showed you on the previous slide schematically? Well, what we did was basically take kind of inspiration from the previous work in our group on protein biophysics and protein interactions. And you can see here three-dimensional crystal structures of protein complexes that we had worked on in terms of or analyzed their assembly using graph theory. So each ball here is a polypeptide chain. Each of these subunits is a polypeptide chain. Here you can see 6 polypeptide chains, 6 proteins in a single 6-fold symmetry, and represented as a graph here. So we had worked on protein complexes and using crystal structures and electron microscopy structures and NMR structures. And we used that as an inspiration to make cell phone. So the cell phone is the database for cell-cell communication. And the reference database, coupled with statistical enrichment of the complementary interactions, allowed us to predict which cells are interacting with each other and what are the complementary receptor-ligand interactions, and what are, sometimes there are multiple subunits, or there are multiple polypeptide chains for a ligand, multiple polypeptide chains for a receptor, and we then use statistical inference to predict which cells are interacting with which ones in the tissue. And so that cell phone database is a resource that we've been developing ever since we published it in that early work in 2018. So I'm going to move basically from cell communication at the maternal-fetal interface, where we learned about tolerance of the fetal cells and uterine tissue restructuring, to another kind of vital organ to our life, which is the heart, which, as you know, sort of pumps the blood around our body. And again, I'm going to focus on what the cell atlas data sets can tell us about cell function by, again, focusing on not just the individual cells, but the niches that they sit in and the interactions between them. And what I want to show here is that, again, like at the placenta deciduous or the placenta uterus interface, in the heart, we have interaction between the cardiac cells, but also the nervous system, and indeed immune and endocrine systems. And the way we tackled this is through a series of really fun collaborative projects with an international consortium of groups in the UK, Germany, and the US. And in this Heart Cell Atlas version 2.0 project, 2 clinician scientists, Kazumasa Kanemaru, who's a Japanese postdoc, and James Cranley, who's a British PhD, finished his PhD now, a clinician scientist, dissected donor hearts from deceased transplant donors into 8 different tissues. And you can see here, we have the muscular walls of the 4 chambers, the septum and the apex, but also the sinoatrial node and the atrioventricular node. And the different cell compartments are shown at a coarse-grained level here. So atrial cardiomyocytes, ventricular cardiomyocytes, endothelial cells, which belong to and neural cells, which make up our vessels, and then fibroblasts, which are sort of the structural cells, and then lymphoid and myeloid cells of the immune system, and also neural cells. And so these are sort of 12 large compartments, but they divide into 75 fine-grained cell states that we described in this work, which is sort of almost 3/4 cells from these 8 different regions. And what you can see in this animation is that, of course, the heart is a muscular tissue on the one hand. On the other hand, it also has its own brain, in the sense that there's an electrical conduction system that coordinates the beating of the atria and the ventricles in a coordinated way. And the rate of that beating is determined by a very small, tiny number of cells in a tiny little tissue called the sinoatrial node that's here, depicted there. And the cells that set the rate of beating of our heart rate, are called pacemaker cells. And these very rare or small number of cells are amongst the only spontaneously firing cells in our body. There's a sort of clock of potassium, calcium channels, and so on that basically have a clock-like mechanism that contributes to this spontaneous firing. Now, of course, as you know, the rate can be modulated, but sort of per se, a priori, it's a spontaneous process that sort of continues from very early, actually, in pregnancy, very early in development, until sort of we have our last heartbeat at the end of life, just continues. And of course, it's a completely independent, autonomic process that we don't think about, just like our breathing. And so what we were super excited about, and this is data from early in the pandemic, where actually transplantation was down in the UK to a minimum, and it was really hard to get these hearts that are, they're declined transplant donor hearts. And James and Kazumasa were able to collect 2 hearts from around the UK, from different centers, London and Newcastle and so on, initially, and then identify the sinoatrial node tissue, which is something that you can't see with the eye, kind of surgically, but working with a cardiac pathologist, Professor Yen Ho at Imperial College, were able to identify the biopsies that contained the nodal regions, and then extract nuclei for single nuclear RNA sequencing, and show that there was this tiny group of about 50 cells that have very high expression of this calcium channel and low expression of this sodium channel. And this calcium channel is basically a marker for pacemaker cells, as opposed to the sodium channel, which is a marker for working cardiomyocytes, so just atrial cardiomyocytes. And so that gave us hope that we had been able to isolate these cells. To analyze, to kind of drill into this in more detail, what James and Kazumasa did was collect all the channels, all the receptors, all the G protein coupled receptors that are known in the human genome, and basically they're sort of plotted along here in the columns, and then the different atrial cardiomyocyte subsets, ventricular cardiomyocyte subsets, and neural cells are shown here in the rows. And you can see this tiny cluster of less than 50 putative pacemaker cells is in this red box. And the pattern of ion channels and receptors is very different from the other cell types that are shown here. And so again, that basically gave us hope that they are indeed, we had indeed identified the pacemaker cells. And then what gave us even more confidence was the spatial transcriptomics analysis of tissue sections from the nodal regions that were identified by Yen. And you can see her annotation here from the histology is that the red region is the node, the epicardium, which is the outer layer of the heart, is in orange, the myocardium, which is the muscular layer of the free walls of the chambers there, and then adipose tissue, and then endocardium here, the inside of the lining of the chamber. And if we then take the pacemaker cell gene expression signature and use a probabilistic modeling to model each of these 50 micron transcriptomic voxels in terms of the weighted combination of cell states, then we see that the pacemaker cells are lighting up in the center of this nodal region that Yen has annotated manually here. And kind of sitting around the edge of these pacemaker cells are fibroblasts and neurons. And so again, we can define computationally kind of a very quantitative sort of definition of tissue microenvironments that's much more fine-grained than what even a trained specialist cardiac histopathologist is able to define under the microscope simply from the tissue morphology. So the genomic analysis of the tissue is revealing not only new cell states, but also new structure of the tissue that hadn't been appreciated before to that level of detail. And if we then validate the pacemaker cells and this tissue microenvironment with microscopy using immunohistochemistry with antibodies for markers for the pacemaker cells, and these glial cells that we had identified for the first time, we can see this really interesting architecture where the glial cells are kind of hugging the pacemaker cells and sort of nourishing them with nutrients and sort of keeping them. Now, glia, as I'm sure you know, are usually lining neurons. So they produce the myelin that electrically insulates and protects our neurons. And these glial cells that we identify here have a different identity, I want to emphasize, to the glial cells that are lining the neurons and that are far away, also in the ganglia. Around the cell bodies. So what we find here is that they are talking to pacemaker cells through glutamatergic signaling, for instance, which wasn't previously appreciated. And we can also define particular stromal cells and a particular matrix around the pacemakers. And what you see here is the innervation of the sympathetic and parasympathetic nervous system. So these are neurons that go through ganglia from your brainstem that are basically tell you to run faster if you see a bear, or relax, basically, if you're at home at the end of the day. And so the heart rate is known to be modulated by cholinergic signaling, which is endocrine signaling. It's known to be modulated by the sympathetic and parasympathetic nervous system. But what was really unexpected and surprising to us was that we discovered the GLP-1 receptor on these pacemaker cells. And the reason that that's surprising is that GLP-1 receptor is the receptor for liraglutide and semaglutide. So Wegovy, Ozempic, Mounjaro, and so on, which are the drugs that regulate hunger and control weight and diabetes and obesity and so on. And although the clinical data from clinical trials reported a 6 beats per minute increase in heart rate upon onset of these drugs in the cohorts, the hypothetical mechanism for that increase in heart rate was through the autonomic nervous system, because of course these peptides are secreted by the gut but are then acting on the hypothalamus in the brain to control our feeling of hunger. And so what was really unexpected was this finding of the receptor in the pacemaker cells themselves, which suggested that there might be a direct action of the drugs on heart rate through that receptor. And the way we discovered that was through a statistical inference of which drugs are acting on which cells. So at that time, we were working on the Genome Campus, where there's the EMBL European Bioinformatics Institute next to the Sanger Institute. And in the EMBL Bioinformatics Institute, there's a beautiful database called ChemBL, which is an open source drug target database, right? So in pharma, basically, we have a lot of proprietary drug and target and side effect and so on databases. This is an open source, completely free database called ChemBL. And we talked to our ChemBL colleagues and downloaded the database and developed a statistical framework for linking the individual drugs to the target molecules and looking for enrichment of targets on cells and predicting basically which cells would be the site of action of drugs. And so that's how Kazumasa and James made this unexpected finding of GLP-1 receptor agonist drugs acting directly on pacemaker cells. And we validated this endocrine mechanism of direct action on pacemaker cells using iPS-derived cardiomyocytes with Michaela Noseda's lab, who's a professor of cardiac biology, cardiology at Imperial College, who did these experiments by ordering the drugs and then culturing the cells in vitro and basically showing a change in beating upon administration of the drugs that was statistically significant versus the control. And so that really supported this direct mechanism versus the other mechanism that had been hypothesized, which is the indirect mechanism of action of these drugs. And since then, this mechanism has been, which I should emphasize that mice don't have this GLP-1 receptor. And so the point that's really, really important to highlight here is that rodent models have, of course, are incredibly powerful to mimic some aspects of human biology and physiology, but not all aspects. So here, there's a difference in the endocrine circuit on our heart rate. Whereas in pigs, basically, this mechanism has been validated since we published this work a couple of years ago by a paper using the porcine system in circulation. And so to summarize this part, basically, I've talked about this new niches for cardiac conduction and interaction with the autonomic nervous system. I've talked about our pipeline called drug to cell for inference of side effects, toxicology, repurposing, and so on. And also discussed both paracrine signaling, like the glutamatergic signaling. Paracrine means close by signaling between adjacent cells. So glutamatergic signaling between glial cells and pacemaker cells, but also endocrine signaling between cells in the intestines and the pacemaker cells. So endocrine signaling can be very far away, basically, essentially across sort of tens of centimeters or even meters, connecting the different systems in our body. And so I want to basically go on to the third story now, which is where we actually make a sort of a further step in cell atlasing and want to model not just individual tissues, so individual cells, individual tissues, but actually an entire organ. And the organ that we're looking at here is the thymus. I'm not sure if you're familiar with the thymus. It's a small organ that's stuck to the heart, sort of between the heart and the lung. It wasn't until the 1960s that it was appreciated that this is an organ of the immune system. Okay, before that, basically, it was just thought the thymus is a bit of fat stuck to the heart because it's quite small. And it was Jacques Miller in London in the 1960s who discovered that it's the site of T cell maturation. So what happens is that hematopoietic stem cells come from the bone marrow, enter the thymus, and then the mature killer, helper, and regulatory and other T cell lineages exit and migrate around the body. And of course, this is basically now also super interesting because of the T cells that are used as drugs. And so the question is, can we actually make T cells, how are T cells made in the body? How can they be mimicked in the dish, and so on? And so the question that we're asking here is, can we actually use this small organ to create a cell atlas of an entire organ? And as I said, the fetal the fetal thymus is tiny, kind of like a pea, and the pediatric thymus is much larger. So the thymus is an interesting organ because it has its maximum size right after birth. And it starts to shrink basically during puberty when our T cell output decreases. And by the time you're my age, your T cell output is about 1% of what it was after birth. So the thymus is also a kind of pacemaker of our life and linked to immunosenescence and the decrease in activity of the immune system. And so the question that we're asking here is, can we learn about the fetal thymus and the pediatric versus the pediatric thymus by modeling multiple modalities together? And the way Nadav Yayon, who was the incredibly talented postdoc who developed this integrative modeling framework, tackled this was by modeling organs and tissues through a nonlinear axis, basically based on landmarks. So the principle here is that you define landmarks, and of course here it's the edge of the thymus versus the cortico-medullary junction, where there's a kind of, you can see here in the histology image, which is the classical kind of staining. And then microscopy is that there's a difference in the outside of the tissue versus the inside of the tissue. So the cortex is much denser and the medulla is lighter. And there's a junction basically in the middle. And so we can just kind of infer the change along this axis by modeling from different landmarks, the edge, the corticomedullary junction, and then there are also other tissues like the Hassel's corpuscles that I won't talk about. And so what that allowed us to do was then take these discrete landmarks and model this axis, which then meant that we could take individual tissue sections. So these are the sections are sort of two-dimensional slices, and then orient ourselves as to where is that tissue section in the three-dimensional organ. So you're basically taking a two-dimensional slice and then orienting yourself. Here you can see the individual slices, and then the different layers of an onion, you're modeling the different layers of each lobe of the thymus from the outside to the inside and getting a kind of rainbow of positioning. And so we discretize these individual layers from the capsule. The capsule is the outside. Then the subcapsular region, which is just kind of inside a lobe, different cortical levels, kind of from the outside, the corticomedullary junction here, and then the different medulla. And then what you can see plotted here in a quantitative framework are basically the early thymic progenitor, which comes from the hematopoietic stem cell, and then different intermediate states. We won't worry about the details. And then finally, the output at the end of this maturation journey are killer and helper T cells, single positive cells, CD4 and CD8 T cells. And what this framework allowed us to do was to model in the fetus and in the pediatric thymus what fraction of cells is at which position. And you can see that the dynamics and this journey from the early thymic progenitor to the single positive cells changes from the fetal thymus to the pediatric thymus. You can see here the early cells are more medullary, and in the pediatric thymus, they're basically both medullary, and then they go out to the cortex. And the intermediate stages basically also change slightly in their dynamics. I'm not going to go into details. I just wanted to kind of give you an idea of how modeling can basically help to integrate both the spatial transcriptomics, the single cell transcriptomics, and imaging, and it kind of opened up a lot of new insights into the maturation of T cells and the decision making between CD4 and CD8 T cells, and also the epithelial cells in the thymus. This is work that was published in November, and the organ axis framework is on GitHub. So we're very keen on always openly sharing software, openly sharing data, and making these data sets and frameworks available for the community. So what's the use, Sarah? What's the use of these atlases? Well, as I mentioned, one of the should I skip that part? It's too detailed. One of the things that could be useful would be to able to synthesize T cells in the dish instead of from a therapeutics point of view. And having the thymus cell atlas, the in vivo thymus cell atlas, really tells us what are the early thymic progenitor cell states and what are the individual points along the journey to make killer and helper T cells. And what's the molecular state of each of these intermediates. And so what that then helps us with, or it essentially sort of gives us the recipe for engineering cells in the dish, in the sense that we know exactly which transcription factor is switched on at which point, what are the signals that the cells are seeing within their tissue environments in the cortex and in the medulla. So what are the growth factors that we should add into the culture to recreate the recipe for making T cells in the dish that they're experiencing in the in vivo thymus. And so what Chen-Chu Suo, who's a pediatric rheumatologist trainee who did her PhD in the lab, did was to set up this artificial thymic organoid culture where you have iPS cells and then make T cells in an in vitro system where you culture the cells on a mouse bone marrow-derived stromal cells that have an engineered delta ligand in them. And this is a protocol that was set up by Gay Crooks at UCLA that we then implemented in the lab. So you have your mouse stromal cells with a human delta ligand, and you culture the iPS cells on top of this layer and achieve hematopoietic induction. So you get hematopoietic stem cells, and then purify the hematopoietic stem cells and culture in an artificial, in another culture for 7 weeks. So it's really not an easy project, I would say, for the PhD, but Chen Chu is an absolute superstar and managed to do this and then characterize the individual time points in the cell culture to track how the cells are maturing in culture, and then use deep learning to project the culture, the cells in culture, onto the in vivo thymic cell atlas and recapitulate the T cell trajectory and identify then also which differences are there using, again, a probabilistic machine learning alignment of the cell culture in the dish, so the T cell development in the dish, relative to the T cell development in vivo. And so what Dinithi Sumanawera, who was a very talented computer science postdoc and machine learning postdoc in the group, did with Chen Chu was to compare the trajectories and then identify which genes are different in the T cell development in vivo versus in vitro. And of course, what we can also do is look at the ligand and receptor expression in vivo versus in vitro. And so what Dinithi and Chen Chu identified was basically when we align in vitro and in vivo T cell trajectories, in vivo we make these mature type 1 innate T cells, whereas in the dish there's a much more immature state that's missing some of the transcription factors, missing some of the receptors, and in particular this map to TNF and NF-kappa B signaling. So what they did was add TNF into the culture and achieve a more mature T cell state where we can now predict what are the other factors that will be needed to get to a fully mature killer T cell state. And we didn't quite push the culture to that point. But just to summarize, basically, the utility of human cell atlasing is also as a recipe or as a sort of engineering blueprint for making cells in vitro. So you can then engineer with computational guidance, basically, what are the factors that need to be added into the dish, what is the CRISPR kind of overexpression of cell intrinsic factors, what are the external factors that need to be added. And just to mention, so this is all published work that's come out in Nature Methods, Nature Biotechnology, and so on over the years. But I want to credit that the team that worked on this was really incredible. Chen Chu is now a trainee clinician, clinical lecturer in the group, completing her clinical training and doing 50% research. Emma is a postdoc at Stanford, and Dinithi is a scientist at GlaxoSmithKline. And of course, we worked with many other members of our lab, with my partner in crime, Mus Hanifa, with whom we've worked together on all the developmental tissues. Also, Menek Clapworthy, who's an incredible immunology and nephrology professor in Cambridge in the clinical school and at Sanger, and our colleague at EBI, John Marioni, who's now at Genentech, and the support teams at Sanger. So I'm going to ask whether I should actually stop, because it's at 7:05, or you want me to go on to another section? Another 10 minutes? There's time. Okay, then I'll go on to one last story. And basically all the previous sections have been talking about really focusing on individual organs. And but what you picked up from the work on the heart is that our physiology is regulated also through signals that come from far away through the endocrine system. And so as I was moving into the clinical school last year, I met a really inspiring scientist called Sadaf Farooqi, who discovered leptin and has worked on melanocortin and these hormones, these peptide hormones that control our hunger and weight. And what we said was, can we team up and assemble a hormone cell atlas of the human body using a data-driven approach to recapitulate the circuitry that controls our physiology through the endocrine system? And in my lab, we have Dr. Lishang Fei, who's an incredibly talented data science and AI/ML postdoc, along with other members of the lab. And in Sadaf's group, Dr. Isabel Huang Dorang is a consultant endocrinologist, clinician scientist, and other members of Sadaf's group who work together on this. And this is really the kind of fun part of moving on to the into the clinical school, where there are all the clinicians around me, is to have these kinds of interactions where, people are thinking very deeply about complex questions related to human physiology, and the connections between the different systems in the body, and also, of course, disease. And so the way we approached this was to integrate over 150 single cell and single nuclear RNA data sequencing data sets from, I think, over 40 different healthy tissues. And this is kind of the data processing pipeline. We take the raw data, integrate metadata about sex, BMI, age, any available clinical parameters, and so on, technical information about the sequencing technologies, use a quality control filter, an automated SC Auto QC, which is our automated filter, and then do a lot of processing and cleaning, and then a unified harmonization of the data with also automatic cell annotation, and followed by manual creation. This really gives us an integrated cross-tissue single-cell atlas. And so what Li-Shang and Isabel are calling this is hormone to cell, with a comprehensive database of hormones, receptors, and then the prediction of the signaling axes. And so hormones, as I'm sure you're aware, include steroids, peptides, proteins, large molecules. And the receptors can be nuclear hormone receptors, GPCRs, or enzyme-linked receptors. And of course, the medical importance of this is for many different diseases, both monogenic and also metabolic. So in our database, there are currently 172 hormones in total. They include manual annotation. We have some enzymes involved in the synthesis and the transport, because, of course, for small molecule hormone steroids or amine-derived, it's not one gene that represents the hormone, but it's the final enzyme of synthesis and the transporter that tells us which cell is sending the signal, right? And it includes neuropeptides, gut peptides, metabolic hormones like adipokines and prostaglandins, and so on. And so we have samples from over 1,000 donors, about half male, female, ages 80 to 90, 10 organ systems, 46 tissues, and 14 million cells. So it's an integrated reference atlas with this hormone to cell framework, and the downstream applications are that we can define the hormone producing cell types, the transcription factors that are regulating hormone-related gene synthesis. We can link to disease genes, and also define the hormone sensing cell types, the long-range networks, and develop a web portal. So I should say this is unpublished work that's not online. It's kind of in progress. It's not finished, but just very exciting work basically from the last year in our group, and I wanted to share this with you just in the last few minutes. So what it allows us to do is to capture classical endocrine cell types. So for instance, in the pituitary, we have these corticotrophic cells in the, and the adrenal glands, which are kind of these classic hormone-producing tissues. We can identify all the key kind of hormone synthesizing cell types. And then from the complete sort of cell and tissue atlas, we can then define where are hormones being produced in the body in a complete unbiased data-driven way based on the complete molecular fingerprint of the cells and superimposing the hormone genes and the synthesis enzymes and the transporters and so on. And then conversely, we can also define the sites of hormone action in a completely unbiased and data-driven way. So what are the tissues that display unique signatures of hormone gene expression? Well, we have the brain and the central nervous system, where we have the hormone genes are kind of shown in the columns here, and the tissues are in the rows, shown here. And we can define basically, like here, the hypothalamus and so on, what are the tissues that have these significant kind of patterns of gene expression. We've got endocrine tissues here, pituitary, thyroid, adrenal, ovary, testis, blah, blah, blah. Adipose also, and gut with kind of characteristic signatures. And the classical kind of genes that we expect are showing up here. prolactin, gonadotrophic hormone. In adipose, we've got leptin, et cetera. And then in terms of the tissues where we have the hormone receptor genes, again, we can see brain very different from the peripheral tissue. So there are sort of distinct patterns between brain and periphery. And then reconstruct basically the classical endocrine axes at cell type resolution. So we've got here the individual neuron, glutamatergic neuron, corticotroph, et cetera, connecting basically the circuits via hormones. So cortisol here, thyroid hormone, androgen, progesterone, so on, connecting the hypothalamus pituitary axis, hypothalamus gonadotrophic axis, hypothalamus pituitary adrenal axis, and so on via these enzymes in the individual cells. Now, of course, we're interested in making sure that we can reconstruct all the classical genes and the classical cells, but what we're super excited about is novel neuroendocrine marker genes. I think there's something funny here. I think this hasn't transferred from Mac. I apologize, but I didn't notice this. But anyway, the main point is that say if we focus on neuroendocrine cell types here, different subsets of specialized cells in the adrenal, but also the intestines, pancreas, stomach, and so on, we can define many different cell types and the hormones and look for top differentially expressed genes in these cells versus other cell types and identify new and surprising neuroendocrine marker genes. And what we can also drill into is the immune system, and which is my kind of sort of special area of interest. You've heard about the placenta and immunity. You've heard about the thymus and the site of T cell production. And so what I was very excited about was when Li Shang kind of showed that plasmacytoid dendritic cells, which is a specialized subtype of innate cells in the blood and an immune system, has expression of secretin that's particularly high after COVID-19 infection. So we were very lucky to be able to study the longitudinal blood kind of single cell response of volunteers infected with COVID-19 during the pandemic in the UK COVID challenge. So it's an experimental medicine challenge where young people between the ages of 20 to 30 were inoculated with the pre-alpha strain of SARS-CoV-2 in late 2020, early 2021. And we were able to follow the blood at day 1, day 3, day 7, day 10, day 14, day 28. And so these are unvaccinated seronegative volunteers. And you can see here the secretin, which is a hormone in these plasmacytoid dendritic cells, is coming up basically day 3 very high in the volunteers with sustained infection. And it's quite high in the abortive infection. And here, different hormone atropin is high in these classical dendritic cells in people who defend themselves from infection. And so what it suggests to us that there may be a sort of cross-tissue metabolic response that's coordinated via the immune system upon viral infection to then secrete hormone and influence basically adipose tissue, kidney, liver, and so on, pancreas, by signaling to cells in these target tissues. And that's, a previously unknown mechanism and unappreciated that we're now following with in vitro assays. So just to summarize this work, which is kind of in flight, it's unfinished, it's somewhat preliminary, so please kind of bear with me, is that by reconstructing basically the networks of hormone production and action at unprecedented, single cell resolution, we can drill into novel discoveries, identify rare endocrine cell types, rare new endocrine cell types with new markers, and provide a resource also for human endocrine and metabolic disease. And so I'm going to summarize basically what I talked about this evening. I've mentioned that we are now in the era of assembling human cell atlas reference data objects, which are being released at data.humancellatlas.org. I've mentioned cellphonedb.org as a resource for receptor-ligand interactions, and the new discoveries that we were able to make at the maternal-fetal interface in first trimester pregnancy. I talked about mapping the cardiac conduction system and identifying drug effects with drug to cell. And then for the thymus, I described mapping a whole organ as a cell atlas for the first time in a quasi 3D, sort of 2.5 dimensional modeling framework with organ axis. And then how we can use genes to genes, which is the pseudotime alignment framework for predicting changes in gene expression between a reference and a query object. So the in vivo versus the in vitro trajectories. And finally, I talked about this ongoing work in progress on the hormone cell atlas to make new discoveries in inter-organ communication. I think in the future, basically, where we want to go is really having a molecular map of the human body in 3D. And what we need to do is integrate the suspension cell data with these small histological spatial modalities with large-scale volumetric reconstructions, like in this hierarchical phase computational tomography or with medical imaging technologies. And we have a program funded by the Canadian Institute for Advanced Research to kind of brainstorm about how can we get to this multimodal atlas of the human body in the long term. These are my disclosures. And I want to stop there, say thank you, and take any questions.
Thank you very much for this lecture, Professor Fleischmann. And
Grazie.
I mean, I was really, when you told me about the CLIP-1 receptor on the pacemaker cell of the heart with an effect, I was thinking, my thought were fly in front of several people they are abusing about the CLIP-1 agonist actually, because they want to lose weight, because they say that it's safe. And all the time I was saying to these people, are you pretty sure? Because, we are at the early of the history and maybe we don't know it. Today you gave me a very important piece of the puzzle to say that maybe my thinker were right. Okay.
I mean, there is a cardiovascular benefit in the long term to losing weight.
Yeah.
So,
but the heart rate, but the heart rate, how changes is increasing.
Yeah.
That's why you don't find it in mice, because you have 600 beats per minute in mice. Yeah. Okay, so we have now the time for our forum. Very exciting lecture and very exciting questions are arriving now from the audience. Is it correct? Yes. Come. Introduce yourself and.
Good evening. I am Tommaso Mario. I'm a medicine student here at Sant'Anna School. And thank you for the amazing lecture. It sounds cliche, but it was truly inspiring. And my question is a bit of career advice. Perhaps you discussed this before, but I couldn't be there. So I'm sorry if I'm asking again.
Feel free.
And so this single cell revolution and also the spatial transcriptomics, these are all things that I find deeply interesting, especially for what concerns the immune system. Now, my question is, as a medicine student with a certain background, how do you think that medicine students can contribute to this kind of research? Because you mentioned several clinician scientists. But when I think, at least now as an outsider, about this kind of research, My first thoughts is that these are more the domains of bioinformaticians and computer scientists. And obviously medical students do not possess the computational know-how to delve at a certain level. So my question is, how do you think that doctors and clinician researchers can truly contribute?
I mean, so I would say all of these projects are huge teamwork. Okay, I hope I tried to kind of emphasize that it's really collaboration between clinicians, basic scientists, computer scientists, mathematicians, et cetera. I've had now 4 clinician scientists, PhD students, and several postdocs, and each one has their own style. I've had an orthopedic surgeon who's basically very dexterous at dissecting with, stereo microscopy, tiny embryo samples. Chenchu, who's actually a brilliant data scientist, pediatric rheumatologist, but actually also brilliant data scientist. So each person kind of finds their own niche and their own combination of skills, what they enjoy, and then what they don't enjoy, somebody else can come along and collaborate and pick up, right? So they're all different kinds of, and you've seen James as a cardiologist, very advanced in training, James Cranley. Kento is sort of transplant surgery. Yeah, so there are many different specialties, and each person kind of finds their own niche and then collaborates, yeah, to complement.
Thank
you. Thank you, Tommaso. Question on science? Yes. First, second, third.
Hello, I'm Salvatore Di Persia, honor student of medicine here in Scuola Superiore Sant'Anna. So first of all, your lesson was very interesting, so thank you very much.
Thank you.
So I have a question as regard the thymoid organoids that you have explained.
Yeah.
So according to your experience and the recent discoveries, what are the possible uses of this new technology in the future research?
Yeah, so basically what we were using it for was also understanding the mechanisms of T-cell development. So the organoids are incredibly powerful for mechanistic understanding of human biology. Because, as we said, like you can't manipulate genetically a human being, but you can manipulate the organoid because you can edit the iPS cells with CRISPR, you can add factors into the culture, like we added the TNF. And the really key mechanistic insight that we got from that culture was that if there's TT cross presentation, so if developing thymocytes present antigen through HLA to each other, you get an innate-like T-cell lineages, which we described for the first time in human. There has been work in the mouse from a Korean group that suggested this mechanism before. So instead of the antigen being presented by thymic epithelial cells, which are the usual cells that are training our T cells in the cortex and the medulla, it was this mechanism of TT cross-presentation, because in the organoid, you don't have epithelial cells. You have the mouse layer, and then you have the thymocytes on top, but actually the mouse cells die off pretty quickly, and then you're left with the developing thymocytes. And so it was, these mechanistic insights, you can isolate cell lineages and study them and so on. That is a very powerful system in the dish. Now, the other obvious application is to make cells for therapeutic purposes. Because at the moment, as you know, the whole CAR T process is expensive and long-winded. And if you could have just T cells off the shelf with different HLA for different histocompatibility for different donors that you can then, simply administer, that would be like a very nice therapeutic kind of product in and of itself. So there are two applications. One is therapeutic thinking, kind of forwards, and the other is gaining mechanistic insights into human biology.
Thank you.
Okay.
And are you designing other organoids?
Well, so we use I mentioned the iPS-derived cardiomyocytes that we did with Michaela Noceda's group. I mean, they're not organoids, but they're iPS-derived cells to show the GLP-1 mechanism and the change in beating rate. We also use gut organoids. So for the intestinal biology, those three.
Thank you very much.
Okay.
The second. Introduce yourself.
Hello, my name is Marcello Zanzi, and I'm a fourth-year molecular biotechnology student at Scuola Normale Superiore. And I was very interested in the parts where you're speaking about the cell atlas and its applications in the use of organoids. I wanted to ask whether or not you have a comprehensive, detailed view of certain developmental processes, and if you're able to link them to the organoids to see what maturity they are able to reach, especially when it comes to cerebral organoids or...
Okay, so we haven't worked specifically on cerebral, but in general, as a principle, this kind of framework, the single cell resolution plus the data science framework for aligning cell types and aligning the trajectories, gives you this very detailed benchmarking of what stage are the cells at. And of course, for the iPS-derived organoids, the cells are often more fetal-like rather than fully mature adult cells. And you have this very quantitative, the opportunity to carry out this very quantitative benchmarking with these kinds of frameworks.
Okay,
yeah.
Thank you.
The third great question. Who stand in the end?
Good evening. I'm Luca Tognari, third-year medical student. I'd like to thank you for your lecture, and it's very impressive. And I'd like to ask you if there is an application of spatial transcriptomics for, discovering new immunological connections and pathways in IBDs, like Crohn's disease or ulcerative colitis.
Yeah, that's a great question. We've worked both on pediatric and adult IBD, and in close collaboration with Matt Zielbauer, who's a pediatric gastroenterologist in Cambridge, and also Fiona Powery, Hom Oleg, and collaborators working on adult IBD in Oxford. And the spatial architecture of the different stages, of course, it changes different stages, and also small intestine versus large intestine and so on is really different. But what it's revealed is a lot of really new insights into the activation of stem cells and the change in this beautiful crypt villus structure to make a kind of much more ugly, disorganized kind of aberrant glandular-like sort of Frankensteinian kind of structures with fibrosis and so on. And the spatial transcriptomics has really given insight into, the identity of the cells, the organization, how the states may develop, clues into that. I should say that also what we've worked on recently, also with Matthias Friedrich and collaborators in Oxford, is actually understanding single cell epigenetics coupled with a spatial organization. So and integrating those different modalities, I think, gives a more complete picture of the development of the pathology.
Thank you.
Yeah.
Okay. Yes. Coraggio, ora è il vostro momento. Dovete fare le domande.
Hi, thank you for the great talk and actually for sharing also the latest bits of research. I really love them. And I'm Martina, I'm a postdoc here in Pisa, and actually I was really thinking once you thought about bringing together all the different modalities that you are going to integrate in an atlas, that especially for hormones, you have a lot of changes throughout life. So of course, you're taking patients who have at least 10 years old, so let's say we forget about puberty, but there's still menopause, for instance, there might be hormonal shifts. How do you think that we can integrate this kind of information when we want to bring that all together into a comprehensive atlas?
Yeah, I think it's a great question. And basically, you know, what we would like to capture in the long term is the whole lifespan, basically during pregnancy, pediatric stages, and then also aging in adult life. And for instance, for the uterus, basically, you know, pre and postmenopausal, Rosa Venter-Tormo has done some beautiful work on, you know, studying the tissues in different stages. And, you know, it's a really big ambition to basically have tissue samples and cohorts from the different decades of life. But that is where we'd like to go in the long term. Yeah. And hopefully that will also give us a deeper understanding of our aging process and tissue aging. Yeah, so great question. Thank you.
Other? Yes.
Hi, nice to meet you, and thank you very much for your lecture. I'm Francesca Ferragina. I'm a third-year medical student here at Scuola Superiore Sant'Anna. And I would like to ask you, how many samples do you need to conduct this type of research? And is it important, and how you handle like inter-individual differences or eventual pathologies?
Yeah. I mean, so the simple answer is you know, the depth and detail of insights is a function of how many samples you have in your tissue collection in all of these studies. And so the initial kind of studies, you know, had small numbers, 10 to 20. And basically that's enough to get kind of the first sort of coarse-grained insights into what are the tissues. So for instance, for the heart, you know, where we're studying the sinoatrial node, they're from deceased transplant donor tissue. It's really hard to, it was fresh tissue and you know, that was really hard to acquire. So we had 5, I think, in the end. But it was enough, you know, to give us those new insights into the pacemaker cells, into the tissue niches, you know, the regulation of the pacemaker cells and so on. But if you want to, for instance, then, you know, as in the previous question, you want to understand how does the heart change in age, male versus female aging and so on. Then, of course, you're going to need a much larger tissue collection to understand those more fine-grained differences. So that's, I hope that gives you a kind of feeling for, you know, the different types of studies. And for healthy disease, of course, again, if you have few samples, you will see a very, you know, the biggest change. But to see, for instance, detailed changes, you need more samples, or to see, you know, different stages of disease and the progression and so on. So that's, but with human tissue research, I mean, a lot of it is a function of the tissue acquisition. It's not like mice where it's just infinite, you know. Okay. Thank you.
Other question? Yes.
Hello, good evening. As you can see, I'm not a student. I'm a pathologist at the University of Pisa. Thanks so much for your great talk. I have a few questions. The first two are scientific questions. The first is, what about making something like you are doing on hormone, but on cytokines, chemokines, and receptor?
We do have that, but they don't travel as far, you know, it's more paracrine.
Okay.
So that's more like a cell phone DB has all the cytokines and chemokines in it. And the reconstruction is, you know, they travel maybe 50 micron or so, but the hormones really travel across the whole body because they're much smaller, you know, as you know. So, yeah.
Okay. And the second is, do you think that the effect of the plasmacytoid dendritic cells you saw on kidney and on other organs that I don't remember, acts also on lungs in the COVID.
I can't remember whether the lungs have the receptor
Okay.
for the secretin.
Okay, but
Yeah,
it
but-
could be interesting
definitely
in a pulmonary infection. Yeah.
Yeah, for sure.
Okay.
Yeah.
And the third question is a more personal question, but I think that it will be interesting for all the people in the room.
Yeah.
How did you manage to be a great scientist and a mother?
So
Because I'm not a great scientist, and I hope to be an acceptable father, but it's very, very difficult to combine.
It is, it's really tough. And I think in our day, kind of, my kids are now 17 and 12. So my first daughter was born almost 20 years ago. There were many fewer policies. And so like 10 days after giving birth, I basically did a PhD interview, because if I didn't do the interview, I wouldn't have had a student the next year, and I could hardly walk, basically. So, that shouldn't be like that, right? One of my colleagues basically, had a child with a hole in the heart, waiting to go in for surgery, and had to, 6 weeks later had to present, after the birth, had to present a defense of her, those things. I hope, this is 20 years ago, right? I hope those things wouldn't happen anymore now. I mean, I think the reason that I've come out healthy and happy, kids are healthy and happy, is just luck, I would say. Very, very fortunate. Also that I was, like the students here, I was at a university, an elite university. I had a very accelerated career, so that by the time my first daughter was born, I had a tenured faculty position. So that I kind of felt unthreatened by whatever happens. And that's, again, luck, And of course, my husband supported, and our family supported, and, so it's circumstances a lot, but it shouldn't be survival of the fittest. It should be that there's a provision and policies that everybody can have a family, have a career, do their research, mothers, fathers, carers, whatever, everybody.
Okay, thanks so much.
Okay. Students, other question? No question. I have a question. So the question is, usually you are investigating the interaction of the cell with the mechanical environment outside the cells. Because if you talk about space transcriptomics and you try to evaluate the interaction even with the mechanical environment, how much change the feasibility of the approach if you want to investigate a tissue that is continuously change the relationship with the environment, like is the beating heart, compared to another tissue that is all the time fixed, like I don't know, the brain, the kidney. The brain, someone say that is beating, but the kidney, no. How much change this relationship with the microenvironment? Because I think about sometimes the modulation of the cytoskeleton, even the trap of some RNA that are entrapped in the cytoskeleton, and I think about Merlin, YAPTAZ, and all this modulation.
Yeah.
So how much is possible to differentiate, stratify the data on the basis of the microenvironment?
So from the transcriptome, which is, the comprehensive data that we have, you can read out, the the extracellular matrix and the differences in the matrix. And that's why I kind of also emphasized for the sinoatrial node, there's a special matrix. I didn't go into it in detail, but there are special fibroblasts and there's a special matrix that's deposited.
Yeah.
You can, of course, infer the difference in the cardiomyocyte, myocyte, smooth muscle, etc.
Yes.
Fibers. So there are certain mechanical or differences in mechanical properties that we can infer from the gene expression pattern based on our knowledge of the genes that are involved. But not everything.
So you relate it to different matrix, you're saying?
Yeah.
So different cells, they have a different matrix.
Yeah.
And what about the shear stress of the interstitial fluids that try to touch the interstitial cells, like some stem cells that are into the tissue? You know that in muscle, you can have a mechanical force from the neighbor cells, but you can have a shear stress of the fluids in the interstitium value that they change during the contraction of the. So how you can control this?
I mean, of course, none of these technologies tell you mechanical load or dynamics because it's all static.
Yeah.
There's no.
But it depends. If you frozen the tissue,
Yeah.
you work on frozen tissue, right? You frozen, you snap frozen the tissue during a loading.
Yeah.
So.
Yeah. I mean, it's actually, you've given me an interesting idea.
Yeah.
Yeah. Because you could try to reconstruct different stages of, let's say, contraction.
Yeah, could be interesting.
Yeah.
Okay, thanks. Question? Okay, so I would like to thank again Professor Teichmann. And now there is the last part that is on the hands of our students.
Yes. Okay, now I'm again going to thank you for your lecture of this evening, this time on behalf of the audience and in particular of the medical students here. And now comes a sort of traditional moment for this event. So we would like to ask you, first of all, to write in this book a thought about this day here at Scuola Superiore Sant'Anna, or a message you would like to leave. Yes.
If you want to read it.
Okay. Yeah. Sure. Sorry. Dear Scuola Superiore Sant'Anna community, it's been an interesting and fun day interacting with the students here and visiting the translational medicine labs and discussing with the teams. I hope you also enjoyed my stories from the land of the Human Cell Atlas project. Till next time, arrivederci, Sarah.
Then here are some gadgets from the school. And last but not least, okay. Here's a certificate that tells that you have been for a day one of our professors here at Scuola Superiore Sant'Anna.
So you are now one of us.
It's an honor.
But now we will take a picture
Yeah, that's very cute. I know. Yeah.
O gumi o amici. Va bene un coltello.
So